We've established that cells can control their hundreds or thousands of chemical reactions by controlling these teams of hundreds or thousands of enzymes. But how do they control the enzymes? Well, there's two major methods that cells use. First, because enzymes are proteins, that means they are coded for in the DNA. There's instructions for making them built into the cell's DNA. The cell can control which enzymes are made at which times by turning the genes for those enzymes on or off. So in other words, the cell gets to decide which workers are present at which times. If it doesn't need to perform a particular chemical reaction, then it can just not make the enzymes that control that reaction. Second, cells have ways of controlling the activity of the enzymes. In other words, they have ways of turning the enzymes on when they need them and turning them off when they don't need them. Let's explore that a little further. One method of controlling enzyme activity that cells have is known as competitive inhibition. In competitive inhibition, a molecule other than the enzyme's substrate will bind with and block the active site. Here, for instance, we have this nice big old purple enzyme, and this molecule has bound to its active site and is blocking it. So because that molecule is in the active site, the natural substrate of the enzyme can't get in there, so the reaction can't happen. Now, cells will naturally produce certain chemicals that will competitively bind with enzymes in order to turn them off at the right times. But lots of toxins and drugs work this way as well. A great example of a toxin that uses competitive inhibition is methanol. Now, ethanol, it starts with an E, that's drinking alcohol, that's booze. And your liver contains enzymes that's capable of breaking it down and detoxifying it for you. If you drink methanol or wood alcohol, on the other hand, that's going to competitively bind to your enzymes in your liver and block them up. And that causes toxins to build up. Drinking methanol can actually pickle your liver from the inside. Now, there are also useful drugs that use competitive inhibition as well. Sometimes enzymes are actually overactive, and competitive inhibitors can be prescribed to shut down enzymatic pathways and solve a problem that way. Competitive inhibitors can also be prescribed to stop bacterial infections. They might bind to certain key bacterial enzymes, and that can help prevent the spread of that infection. Another means of controlling enzyme activity is known as non-competitive inhibition, or allosteric inhibition. In this type of regulation, a little regulatory molecule will bind with a site away from the active site, known as an allosteric site. When it binds to this regulatory site, it forces the enzyme to change its shape, its conformation, and that is either going to make it better able to grab the substrate, or in this case, not able to bind with the substrate at all. So depending on the regulatory molecule in the enzyme, it can either increase the enzyme's activity or decrease the enzyme's activity. A great example of non-competitive inhibition happens with your red blood cells. Now, it's the job of the hemoglobin inside your red blood cells to bind to and carry oxygen throughout your body. When oxygen binds with the binding sites on red blood cells, it actually improves their ability, their affinity for oxygen. So the more oxygen that binds to them, typically the more oxygen they can carry. Now, if your red blood cells happen to bump into carbon monoxide, that silent odorless gas that's produced in car exhaust, for instance, carbon monoxide will bind with hemoglobin and will block it up. So when your red blood cells bind to carbon monoxide, they can carry less oxygen. So it makes them less able to do their job.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.
Sometimes metabolic pathways are sort of auto-regulating or self-regulating. Some of them use something known as feedback inhibition. In feedback inhibition, the end product of a metabolic pathway affects one of the enzymes used early in the pathway, shutting it down. Let me give you an example. Let's say we have a pathway here and we start with pink molecule, and what we want to make is lots of this blue molecule down here at the end. So the pathway begins, the enzymes start doing their jobs, and we start making a lot of blue. As this blue molecule builds up in higher and higher concentrations, it's going to begin affecting the activity of this first enzyme in the pathway, and it tends to shut it off. Now, if this first enzyme isn't working and producing intermediate product A, the rest of the pathway can't do its job either. So eventually, enzyme A is going to stop working and the whole pathway is going to shut down. Now, why would you want to do this? Well, feedback inhibition ensures that as this blue molecule builds up to the proper concentrations, that the cell isn't wasting a lot of time and energy making even more blue molecule. When the proper levels of blue molecule are reached, the pathway shuts down. As the blue molecule is used up, enzyme 1 can reactivate and the entire pathway can start working again, so you can make more blue molecule.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Other factors can affect how enzymes behave. First, let's talk about some little helper molecules. Helper molecules assist enzymes in speeding up their reactions, and enzymes typically don't work very well without them. Inorganic helpers we call cofactors. These can include things like metals such as copper, zinc, iron, and other trace elements. Organic molecules that assist enzymes are known as coenzymes, and molecules that we classify as vitamins fall in this category. So another way of controlling the activity of enzymes is to control the concentrations of cofactors and coenzymes that are present. If an enzyme requires zinc to work, for instance, and zinc is not present, the enzyme is essentially shut down. Different enzymes work best at different pHs, and this kind of depends on where they live in quotation marks. Here's two examples. Pepsin is an enzyme that breaks down proteins in the stomach. Remember PEP for proteins? Now notice its optimal activity. It works best at a pH of around 2, And that's because it exists in this sort of sea of hydrochloric acid inside the stomach. Now, salivary amylase is an enzyme that breaks down sugars in your saliva, and its optimal pH is around 7. That's because the saliva has a more neutral pH. If I were to take pepsin and toss it into the saliva, it's not going to work very well. And if I took amylase and tossed it into the stomach, it's not going to work very well there either. So each particular enzyme has an optimal pH, depending on where it exists. It's also important to note that really extreme pHs, either really acidic or really basic pHs, can actually denature enzymes. Remember that enzymes are proteins, and proteins are sensitive to having all their little internal bonds broken. So exposing it to an extreme acid or an extreme base can cause the protein to lose its shape, and it can't do its job if it doesn't have its three-dimensional shape. Enzymes also have temperature ranges that they prefer. The enzymes that work inside our bodies, for instance, really like body temperature, so that's 37 degrees Celsius. When the temperature goes down, enzymes may not have enough kinetic energy, enough motion, to perform the reactions. As the temperature rises, enzymes will gain kinetic energy, so for a while they can actually perform their reactions a little bit faster. But at a certain point, the temperature is going to be so high that the enzymes will begin to fall apart. Remember that enzymes are proteins, and high heat can actually destroy the shape of a protein, denature it, and thus it can't perform its function. This is the reason that having a high sustained fever can be really dangerous. If you have a fever for a really long time, eventually the enzymes in your brain and in your blood can begin to denature, and that can cause other complications. Another factor that can affect the rate of enzyme activity is the concentration of the substrate. It makes sense that if there's just a little bit of substrate floating around for the enzyme to work on, that those enzymes aren't going to produce very much product. Certainly they're not going to produce product very quickly. If you increase the amount of substrate floating around, then the enzymes can work a little faster. They can make more product a little bit quicker. This direct relationship between substrate concentration and the increasing rate of reaction is only true up to a point, though. Let's talk about that some more. In general, the addition of more substrate to a solution will also increase the rate at which enzymes make product. That's only true up to a point, however, and that point is called the Vmax. The Vmax is the point at which all of the enzymes are already bound to substrates. So in other words, they're working as fast as they can. At this point, adding more substrate isn't going to speed up the reaction anymore, because they're already as busy as they're going to be. If you want to speed up the rate of reaction beyond the Vmax, you can either add a little bit of heat to the situation, or you can add more enzyme. In other words, you can add more workers to the situation instead of more ingredient.
Right, girls, listen carefully. This is the wrapping department.
Yes, ma'am.
Now, the candy will pass by on this conveyor belt and continue into the next room where the girls will pack it. Now, your job is to take each piece of candy and wrap it in one of these papers and then put it back on the belt. You understand?
Yes, sir. Yes, ma'am.
Let her roll! Let her roll! Well, wait here. Somebody's asleep at the switch.
What are you doing up here? I thought you were downstairs boxing chocolates.
Oh, they kicked me out of there fast.
Why?
I kept pinching them to see what kind they were. This is the 4th department I've been in.
Oh, I didn't do so well either.
All right, girls. Now, this is your last chance. If one piece of candy gets past you and into the packing room unwrapped, you're fired.
Yes, ma'am.
Let her roll!
Oh, this is easier. Yeah, we can handle this, okay? Listen, Ethel, I think there's... I think we're fighting a losing game. Here she comes.
Fine, you're doing splendidly. Speed it up a little!
All right, you're finished with this chapter. You should feel as proud as an enzyme who has been reunited with his long-lost substrate. Make sure you complete your guided notes for this chapter and bring them to class, because we're going to go over them.